Hey, Hammy here, back with part 2 of 5.2. In this video, we want to take a look at mitosis. Okay, last video we talked about the chromosomes and kind of the human chromosome and how it contains our genetic information. And it kind of looks like that X thing. Okay, in this video, we want to look at the mitotic stage of the cell cycle. So how does the cell actually divide its nucleus, which is mitosis, and the cell cytoplasm itself divide, which is cytokinesis. Okay, again, just a reminder, when we talk about the actual process of mitosis, we are only referring to the cell dividing up the DNA within the nucleus, okay, not the actual cell dividing itself, but we're just referring to in mitosis dividing up the DNA so each daughter cell gets a complete set of the chromosomes. We usually talk about it in 4 phases. Okay, the actual phases of mitosis, remember, are PMAT, prophase, metaphase, anaphase, telophase. In the first stage, prophase, okay, this is the longest phase in mitosis, and we'll see that in lab when we begin kind of counting cells in the onion root tip. In the cell, it really begins to sort of break down its normal structures here and begin to prepare for cell division. So you see the chromatin, which is normally long, stretched out, real thin DNA, that begins to like coil up. And I showed you in the last video kind of how DNA is wound around these proteins called histones. And then those things coil up, and it goes from kind of the stretched out, usable form into this condensed form called a chromosome. Okay, that happens in this first stage here in prophase. The nuclear membrane begins to break down. So nuclear membrane is the membrane around the nucleus, and you can see here how it's kind of shown as a dashed line, like it's slowly kind of fading away. Okay, you also have these things called centrioles. Okay, these centrioles often are at a right angle. They look like 2 little bars or pipes that are at a right angle to each other. They will form the anchors during cell division. So you'll see them in the cell here. They're kind of moving to opposite poles or opposite ends of the cell. And then they begin to form these spindle fibers. And here you can see these branching out spindle fibers that will kind of anchor it in and kind of help stretch the cell and also grab the chromosomes and pull them apart. Okay, so this is all happening in prophase. If we look at kind of what a typical onion root tip cell that we'll look at in lab as well. In prophase, you'll see it kind of just looks like a normal rectangular cell. You can kind of see where the nucleus was, kind of around here. But you'll notice that instead of a nice even shaded nucleus, you'll see that the nucleus, the DNA is all clumpy. Instead of even shade, these dark kind of looks like yarn or threads or kind of long clumpy strands. This is a signal that the cell has begun prophase and is ready to start dividing. The next stage is metaphase. Okay, metaphase is an easy one to tell when we're looking at cells in a microscope, or if you just have like figures you have to identify for a test, because in this the spindle fibers reach out and then they attach to the centromere of each chromosome. Okay, so you remember our chromosome were kind of these X-shaped sister chromatids. Okay, the centromere is what held the 2 halves together right in the middle. Okay, the spindle fibers are going to reach out from each end and attach onto those centromeres. So here you see the centrioles have moved to the poles of the cell. The spindle fibers are going to reach out and they're going to grab hold on both sides of that chromosome. Okay, that imaginary line where they're sort of lined up is called the metaphase plate. Over here is, again, here's our onion root tip cell over here. And you can see kind of all these squiggly lines. They're starting to sort of line up. Well, that's not a perfectly straight line like the figure. Yeah, I know, but this is a real cell, and there's like strings and ends and tips of chromosomes, and you're looking down above, and it's 3-dimensional. But you can kind of tell that everything is sort of right in the middle of this cell there. Okay, so this is an easy one to tell when we look at microscopes or when we look at figures. Step 3, okay, step 3 is anaphase. And in anaphase, this is the muscle, okay, where the spindle fibers begin to begin to shorten, okay? Excuse me. And you notice that each spindle fiber is attached now to one of the sister chromatids. Remember, they are two exactly the same pieces of DNA, okay? And it's going to pull one direction in the cell, and the other centriole and spindle fiber is going to pull the other one the opposite way in the cell. Okay, so anaphase, by the end of anaphase, okay, each end or each side of the cell will now have a complete set of chromosomes. Remember, what's the whole goal of mitosis? Divide up the DNA in the nucleus. Okay, so they line them all up in the center. The spindle fibers attach in anaphase. Now they are pulled to the opposite ends, which will become the daughter cells. Over here in our example of the onion, again, here it's easy. This one is also easy to tell because the two sides, there's a space in the middle where they're being drug apart. The last and final stage is telophase. Okay, this is kind of, I like to think of it as being opposite of prophase. So now the DNA has been drugged, okay, right here and right here has been drugged to opposite ends of the cell. And you'll see that the nuclear membrane, this dotted line, dashed line, is starting to reform there. Okay, so the nucleus is reforming. The spindle fibers begin to break down. Okay, and you really are starting to have kind of the situation set up where now it's actually overlaps with cytokinesis, where the cytoplasm will kind of start to divide this thing into two separate cells. And when we look at it in the onion root tips, we can see that now there's that space like anaphase, but they're kind of clumped back up together, and we can begin to see, because it is a plant, we'll talk on the next slide here in a second, it's beginning to build that cell wall between them, and the cell has been stretched apart. It'll be wider than the other cells around it. You'll see two clumps kind of, they've moved. Okay, understand that mitosis, it's not like you jump from a step to a step to a step like you're jumping down the stairs. This is a continual process. So some books even talk about pro-metaphase or early metaphase, late anaphase. Okay, this is all one step to the next, and it all kind of runs together. It's one continuous phase. Okay, so it's important to understand that. So sometimes anaphase and telophase can be a little tricky to tell apart. Okay, this figure gives us just kind of an idea showing the chromosomes you get from your mom and your dad and how they're in your cell and the different stages. Okay, so most of the cell cycle, okay, G1, S, and G2. Okay, remember that's all lumped together in interphase where it's growing. When it actually divides, okay, mitosis, prophase, nucleus disappears. Here you see these would be your homologous chromosomes, and these would be a pair of homologous chromosomes. The lighter colored ones from mom, the darker green ones from dad. Okay, metaphase, remember I said that's an easy one because they line up here in the center. Okay, spindle fibers attach. Anaphase, they get pulled apart. Okay, now notice one sister chromatid one way, one the other way. Okay? And then telophase, nucleus reforms, spindle fibers disappear, and then also cytokinesis begins, in which the cell starts to divide into two actual daughter cells. Okay, but you'll notice that when you started the cell, you had 1, 2, 3, 4 chromosomes. Notice when we end, we have 1, 2, 3, 4. Okay, we have 4 chromosomes. So they're genetically identical. Well, you say, well, but there's only one sister chromatid, not 2. Well, when this goes through the S phase of the cell cycle, it will duplicate those and it'll be back to kind of that X shape that we recognize. Finally, kind of overlapping with telophase is the actual division of the cytoplasm in the cell into two daughter cells called cytokinesis. Okay, this is the final stage where the cell actually divides or splits into two daughter cells. Now notice there's a difference between animal and plant cells, and that's the reason I wanted to take a look at this figure that's in your notes. in an animal cell, this would be animal cell over here. Remember, animal cells do not have a cell wall like plant cells do. So what can happen is they get a little microfilament ring. It's like taking your belt and putting it around a balloon and just pulling it real tight. What will happen? The balloon will pinch in the middle and eventually pinch into two. And then it seals off the two cell membranes here in the middle. And then pop, you've got your two daughter cells. Okay. In a plant cell, it's a little harder to do because it has that rigid cell wall around it. So what it does is it builds us what we call a cell plate. Okay, the two new cells begin to lay down. You see these little molecules, these little dots here. It begins to lay down cellulose and other other organic molecules here that will build that cell wall between the two new plant cells. Okay, we call it the middle lamellae, and it just forms a new cell wall between them. And then pretty soon, once it's finally finished, then you've got 1, 2 daughter cells over here as well. So just a little bit different. Animal cells can just pinch into 2. Plant cells, they actually have to kind of build a new cell wall between the 2. Okay, so again, that was a lot. Okay, that was a lot of information on all those stages and cytokinesis. Please go back and rewatch this. I will most likely go over this in class as well. So make sure you fill in your notes and add things to help you understand this.